So continuing our discussion of microorganisms, we're going to talk about viruses. So viruses, of course, have been bugging us for a long time, and they've pretty much ruined our lives for the last couple of years. So, or particular ones anyway. And so let's talk about them and figure out how are they different from bacteria and why they're so mean to us. So the learning objectives, some of them carry over from the previous lecture, but listing the different types of microorganisms and being able to describe and distinguish the features of each type. Which types of microorganisms are obligate parasites requiring a host to survive? Hint, viruses. Viruses are obligate parasites. I want you to be able to list the medical treatments for viral infections. And do antibiotics kill viruses? Why or not? Why or why not? So viruses are small, obligate intracellular parasites, meaning they need you to live. They need you to reproduce. They don't have the mechanisms in their own structure to reproduce themselves, and so they need to borrow your cells in order to do it. So they're usually they're really common pathogens that cause diseases in humans. They need living tissue to replicate on, and that complicates laboratory procedures to grow or test viruses, because you can't just grow them in culture like you can bacteria. You need cell cultures, and you need the right cell cultures. So viruses are specific for the types of cells that they are attracted to and the types of cells that they need to replicate themselves. So before it has found its cell that it's going to parasite onto, so it's extracellular, the virus particle is called a virion. It consists of a protein coat or a capsid and a core of either DNA or RNA. So there is one of the green and white table in the book that I like so much, that lists common viral diseases, what type of virus it is, whether it's an RNA or DNA, and an example of the disease that it causes. The protein coat and the virus itself can change and mutate quickly, which is one of the reasons why it's really hard to pin these guys down. They come in lots of shapes and sizes, and the nucleic acid content and the form is how they classify viruses. So a virus that contains RNA only as its nucleic acid is called a retrovirus, and the retrovirus has to enter the human cell or the cell that it's preying on. It codes for its own enzyme called reverse transcriptase to translate its RNA into DNA, and then it uses your cellular mechanisms to translate its DNA into its own proteins to replicate itself. So some RNA containing viruses contain reverse transcriptase, and some have to use yours. So viruses are highly adaptable and highly dangerous. So there are lots of different shapes. You can have round ones. This is just, obviously an artist's rendition of them. The one with all little spikes on the outside is a coronavirus. So COVID-19 is caused by a particular coronavirus, which we'll talk about. But there are lots of other kinds of viruses, but they all have that protein coat, the capsid. They all have some kind of envelope to hold their DNA or RNA, and some way to attach to the cells so it can inject its DNA or RNA into your cell. So in an active viral infection, the virus attaches to the host cell it injects or somehow gets its genetic material into your cell, the viral DNA or RNA takes control of the cell, and it uses the cell to synthesize the viral proteins and nucleic acids. Then new viruses are assembled in the cytoplasm of the cell, and then the viruses are released by lysis of the host cell or budding from the host cell membrane, and they effectively kill the cell upon leaving it. So if you have a virus that targets certain cells, the symptoms that you end up having are going to be involved with those cells. So the first form of COVID-19 was targeting lung cells and respiratory cells, and that's why people had those respiratory symptoms. As it starts to mutate, it can start to target other cells. So frequently one type of virus exists in lots of similar forms or strains, and viruses tend to mutate or change slightly during replication. So like the cold or flu viruses, there are lots of different strains that affect humans and animals. So it's the mixtures in the DNA of the virus can change rapidly, and that's one of the ways they try to defeat our measures to take care of them. So certain intracellular viruses can also alter the host DNA chromosomes, leading to development of malignant cells or cancer, like human papillomavirus is a cancer-causing virus. There's a vaccine available for human papillomavirus now, which is great. And vaccines are one of the ways that we can combat viruses. So this is viral replication. The virus is just floating along, attaches to the host cell, it penetrates the host cell's cell membrane, it uncoats its viral DNA or RNA, and it takes control of the host cell's DNA. So then the host cell synthesizes the viral components, assembles new viruses, and then releases them. It bursts through the cell, usually killing the host cell, and then those little replicated viruses are off to infect other cells in the region. So not a fun thing. So when a virus enters a cell as with active infection, it's producing its viral proteins, and sometimes it inserts its genetic material into the genetic material of the host cell. Sometimes viral proteins are produced and they stimulate an immune response and destruction of the host cell. Sometimes a virus can reproduce actively if the immune system is depressed. So herpes virus is an example of a latent viral infection that just sort of hangs out and waits till your immune system's down, and then it expresses itself. So unfortunately, those guys tend to stay with us for a long time. Another latent viral infection that a lot of us have is the varicella zoster, the chickenpox virus. We got chickenpox as little kids. We survived it. The virus stayed in our nerve cells, and then later on, it manifests itself as shingles, which is very painful. And so there are different shingles vaccines now too. So even if you've had, and there are chickenpox vaccines as well. When I was a kid, there weren't chickenpox vaccines, and you just had to get it and deal with it. And luckily I got it when I was too young to remember. So that's one thing, but you can get tested. You can get a blood test to find out if you still have antibodies to chickenpox, and you do. So when the hospital where I work, when I first started working there, you had to show proof of several different vaccinations. And if you were old enough, like I am, to have grown up before the vaccinations were available, they made you get blood tests to test whether you had antibodies to chickenpox and some of the other things. So so you can test for that. But that also means that if you have the antibodies to the chickenpox, to varicella zoster, then you were exposed to it as a child, and it means you could have shingles as an adult. So getting a shingles vaccine is a good way to not get shingles. So antiviral drugs, vaccines are one way to fight viruses. So vaccines, we'll talk a little bit more about how they work in the immune chapter, which is the next chapter, but they are preventative of getting the virus. Antiviral drugs are what you take after you get the virus. So antiviral drugs can act different ways. One of the ways they can block entry into the host cell, they can inhibit gene expression, they can inhibit the assembly of the virus in some manner. So antiviral drugs usually have to be specific to the virus that they're trying to fight. So currently there are antiviral drugs available for flu viruses. There is now an antiviral drug available for COVID, which is called Paxlovid, and it's usually prescribed to people who have high risk for complications with COVID. So it's someone's been vaccinated, they get a mild version of COVID, and if they have high risk factors for other things, they can take the antiviral to lower the viral load in their body and hopefully head off the complications.